All right, today we're going to dive deep and deconstruct a really crucial lesson from the Inquiry Hub biology curriculum. We're talking about the one that masterfully connects the dots between natural selection in a population and the much bigger story of how new species emerge.
This is the big challenge for us as biology teachers, right? How do we help students make that huge leap from seeing how one population changes over time to understanding how a whole new species can be born? Well, we're going to break down exactly how Lesson 25, What Other Juncos Do We Find in North America, is designed to build that bridge.
But before we get into the nitty-gritty of this one lesson, we've got to zoom out. To really get why this lesson works so well, you have to see where it fits in the beautiful narrative arc of the entire evolution unit.
So the unit is really cleverly split into two big parts, or bends. Bend 1 was all about the case of Addie and her bacterial infection. It was a perfect way to ground students in the basics of natural selection, but in a very simple system. Now, in Bend 2, the lesson completely changes the game. We're moving up to complex, sexually reproducing animals, the Juncos, and tackling a much bigger question. Now, coming into Lesson 25, our students aren't starting from scratch. They've already done a ton of great investigative work. They've established that the Juncos on the UCSD campus are different from the ones up in the mountains. They look different, they act different, and most importantly, they get that these differences are heritable adaptations, all driven by the different pressures in their environments. And this is the pivot. Lesson 25 intentionally flips the whole script. We're moving beyond just describing how populations are different. The new question, the big question, becomes, wait a minute, are we watching them become two completely different species? It's a huge step up, and this lesson is the bridge that gets them there.
So, let's dig into the first part of the lesson and see how it introduces this really compelling new puzzle for the students.
The lesson kicks off by throwing a wrench right into a super common student assumption. It just floods them with images showing the incredible variety of Juncos across North America and basically asks, so, all these birds look different. Does that automatically make them different species? And you can see why this is such a great challenge. Students see photos just like these. The slate-colored Junco, the Oregon Junco, the gray-headed Junco. I mean, they look so incredibly different. This visual evidence just forces them to grapple with the fact that defining a species by looks alone is going to be really tricky.
And this is just a brilliant pedagogical move, isn't it? It connects right back to what they learned about the bacteria in Bend 1. They see that this concept of variation, this core pillar of evolution, isn't just some weird thing happening with microbes. It's happening on a continental scale with these birds. It just makes the concept so much more real and powerful.
So the whole strategy here is to create a problem for the students. You're intentionally guiding them to a place where they realize that their current tool, just looking at the birds, isn't good enough. This creates a genuine need, a real curiosity for a better, more scientific way to figure this out.
And once they're practically begging for that better tool, the lesson delivers this new, powerful way of thinking about what a species even is.
And here it is, the core idea of the lesson, the biological species concept. This just completely shifts their focus. It's not about what they look like anymore. It all comes down to a much more fundamental question. Can they breed with each other in the wild and produce healthy offspring that can also reproduce? That becomes the new gold standard.
But they don't just memorize the definition, of course. They immediately put it to work. They get a map showing the breeding ranges for all those different-looking Juncos. Their job is to predict where these populations might actually overlap and interbreed. And as they do this, they discover these things called hybrid zones, which is concrete proof that, despite looking so different, these birds are, in fact, interbreeding.
And that right there is the key instructional payoff. Students have to abandon their first, simpler idea that was just based on looks. They are forced to reevaluate all the evidence through this new lens of the biological species concept, and they learned a huge lesson. Looks can be deceiving.
But the lesson doesn't just stop there and wrap everything up in a neat little bow. And what I think is a brilliant move, it concludes not with a tidy answer, but with an even sharper, more compelling question. Okay, so at this point, the whole class is on the same page. They've used the biological species concept and the map evidence to establish that all those diverse-looking Juncos across the continent, they're all members of the same species. Case closed, right?
And then, bam, the lesson brings it all right back home. It takes this huge continental concept and focuses it like a laser on the original case study. Okay, great, it asks, but what about our Juncos, the ones right here? Do they interbreed? All of a sudden, the stakes feel really personal and immediate again.
And this is the moment where students realize there's a huge gap in their evidence. They've watched the bird's behavior, they've measured their traits, but none of that is enough to answer this new, more sophisticated question. How can we really know if they're still interbreeding or if they've already started that journey of splitting into two separate species?
And that sets up the absolute perfect cliffhanger. The lesson has created a genuine intellectual need for a totally new kind of evidence. To truly answer the question for the UCSD and mountain juncos, we have to go deeper than what we can see. We have to look at their DNA. This creates this fantastic narrative tension that leads them right into the next lessons on molecular evidence, leaving them with a real driving question for their next investigation. It's just a perfect setup.